The organism I selected for our project is Borrelia burgdorferi. Borrelia burgdorferi is a gram-negative spirochete, meaning it is long and helical, as you can see in the picture. One of the papers that I summarized for the project studied the flagella and its function in the organism. The study found that the flagellum was responsible for mobility and cell morphology, which is fairly rare. Typically, the flagellum is responsible for mobility, but not always cell morphology. They were able to determine this function by inactivating a periplasmic flagellar filament protein called FLAB, F-L-A-B. Once the organism no longer had the flagella, it had a rod morphology, so the actual shape of the cell changed. This caused the bacteria to no longer be mobile and therefore determined that the flagella is important for multiple reasons. One, for mobility, and two, for the cell structure. This organism lives in the midgut and saliva of deer ticks. As you can see in this picture, it would be difficult to tell which ticks may or may not carry the organism with just the naked eye. B. burgdorferi is the cause of Lyme disease, which is a zoonotic disease, meaning it can infect humans and animals via tick bite, but cannot be shared from mammal to mammal. You can see here in the life cycle of the tick, it begins as an egg, turns into a larva where it can feed on small mammals that may also be a host for B. borrelia, which can then translate into the adult tick, which can feed on larger mammals such as the deer, raccoon, human, and dog. So how is B. burgdorferi transmitted from the tick to the actual host? Once the tick is attached to the host, changes on the outer surface proteins called the OSP proteins start to begin. The first protein expressed is the OSP-A protein, which allows the spirochete to move from the tick into the host. The protein is expressed to localize the spirochete in the midgut of the tick, meaning that it's kind of in place, not necessarily ready to move until the OSP-A protein reaches it, and it kind of detaches itself in the gut. All of this starts to begin when the tick starts to ingest a blood meal. At that point, OSP-C is expressed to promote movement of the host, movement into the host. So at that point is when it moves down from the tick gut and into the actual host animal. Once inside the host, the organism will shed the OSP such that it's not detected by the host's immune system. At this point, it disguises itself with a protein VLSE to avoid elimination by the host immune system. Despite its efforts to go unavoided, at this point is where the immune system can start to detect C6 peptides, which are part of the VLSE kind of outer coating. And the immune system can start making antibodies. Antibodies can be detected within 3 to 4 weeks post-infection in the host mammal. VLSE is expressed only when inside an actual mammalian host. It's not expressed inside the tick. Therefore, detection of VLSE or C6 inside a host animal can be distinctive for an active infection and not only exposure. Over weeks and months, C6 antibodies will start to increase and symptoms may start to occur. Treatment for active infections include oral antibiotics. The duration is usually 21 to 28 days and usually covers the infection. Once treated, the C6 antibodies will begin to decrease. Something that I found very interesting with this project is the comparison of animal health to human health. I find this interesting because my background for the past 12 years has been in animal health. You'll see here on this slide, there's a white little snap test that is part of the recommended annual visit for a canine. So it tests for heartworm disease, which is something you might be a little more familiar with, and then also 3 tick-borne disease, including Borrelia burgdorferi, which is Lyme disease. So it's part of just annual detection for a dog. This dog here in the picture is my dog Rocket, who unfortunately I call him a retired hunting dog. I found him on the side of the road many years ago. But when I found him from his exposure from the elements outside here in eastern North Carolina, he did test positive for Borrelia burgdorferi, and we put him on a course of 28 days of doxycycline, and luckily he's never shown any symptoms over the years. But so that kind of covers for canines. It's something that is monitored for and detected fairly regularly. In humans, that doesn't necessarily seem to be the case. You'll see here on the right that it says there's no reliable test, and fewer than 50% of patients even remember the tick bite happening. A lot of times it takes quite a bit of time of being sick and showing symptoms and process of elimination through many, many tests before there's a diagnosis of actual Lyme disease. So that makes a huge difference from detecting it just on an annual screening. Some of the Lyme disease symptoms for early Lyme disease include fatigue, headache, rash, fever, sweats, chills, muscle pain, joint pain, neck pain, and sleeping issues. Again, most of the time early Lyme disease can be resolved. Chronic Lyme disease can be much more involved. As you can see, it can start to affect the heart and cognitive abilities. Here's another slide that I found to be very interesting. As you can see, it's a map of the United States, and it shows the forecasted prevalence of Lyme disease. This is forecasted for canines. As you can see down in the bottom corner, it's by the Companion Animal Parasite Council, and you'll see that the upper right-hand part of the United States is where it is the most prevalent, but both human health and animal health are finding that it is increasing, and it's going into states and counties that have never seen it before. You can see eastern North Carolina, where we are, is fairly yellow. It is something that is detected in both humans and animals. But one of the articles that I read as I was preparing for this project is that they are actually basing a lot of human research on what they have already found with animal research, because there are routine screenings and a lot of unsymptomatic cases that are detected and resolved before they even increase into major sickness. They're using a lot of that data to predict where are we seeing more instances of Lyme disease, where do we need to be more concerned for human health, because obviously if these dogs are being exposed, they're most of the time going wherever their human has been. So it's certainly, as it increases for the animals, the risk increases for us. So I think that's cool, the way that they can put the two different sides of medicine together and complement each other. So awareness and prevention. So how can you protect yourself from Lyme disease? So for the human to get Lyme disease, obviously the tick has to attach to you. So be cautious of your surroundings. If you're going into wooded areas or fields and such, maybe wear clothing that protects your skin, or if you have been outside in those areas, at least make sure that you're checking your body, make sure that you do not see any ticks or have any ticks attached. If you have a tick attached to you, be sure to take it off and monitor the area. If you have any redness or any circles develop in that area, you certainly would want to contact your physician. Also, protect your animals. So your animals can bring ticks to you, unfortunately. So ways that you can protect your animals is put them on monthly tick prevention. There's certainly many different products for that. And then, an additional way to protect your canine friends, or there are Lyme vaccines for dogs. So, after learning a little bit more through this project, I did research and look if there's Lyme vaccines for humans. At one point there were, but with the vaccination controversies that have arose, it was removed from the market. They no longer have a Lyme vaccine available, even if you wanted one. So I think the main thing for humans is just awareness and detection. So certainly, keeping a check on yourself, and if you have an area concern, going to your doctor and letting them know about it so that they can prescribe you the proper antibiotics and medications before symptoms or anything occur. So hopefully you learned something new from this project that you can protect yourself or your furry friends. So thanks.